1. Weeds. Weeds are plants that are unwanted in a given situation and may be harmful, dangerous or economically detrimental. Weeds are a serious threat to primary production and biodiversity. They reduce farm and forest productivity, displace native species and contribute significantly to land and water degradation. The costs of weeds to the natural environment are also high, with weed invasion being ranked second only to habitat loss in causing biodiversity decline. Weed is a plant growing out of place and time. They are unwanted, not useful, persistent and prolific, effectively competing with the beneficial and desirable crop plants for space, nutrients, sunlight and water, interfere with agricultural operations and thereby reducing the yield and quality of produce. Weeds are unwanted and undeserved plants that interfere with the utilization of the land and water resources and thus adversely affect crop production and human welfare. Thus a plant out of its place or a plant growing where it is not desired at that time is a weed. This definition was given by Buchholzer in 1967. E.g. bajra is a weed in pulse, pulse is a weed in bajra, tomato is a weed in brinjal field. Characteristics of weeds. Weeds are also like other plants but have special characteristics that tend to put them in the category of unwanted plants. Most of the weeds, especially annuals, produce enormous quantity of seeds, e.g. wild oats, Avena pachua, produces 250 seeds per plant, whereas wild amaranth, Amaranthus viridis, produces nearly 11 million seeds. It has been observed that among 61 perennial weeds, the average seed production capacity was 26,500 per plant. Weeds have the capacity to withstand adverse conditions in the field, because they can modify their seed production and growth according to the availability of moisture and temperature. They can germinate under adverse soil moisture conditions, have short period of plant growth, generally grow faster rate and produce seed earlier than most of the crops growing in association. Weed seeds remain viable for longer period without losing their viability, e.g. annual meadow grass, Poa annua, and scarlet pimpernel, Anagales arvensis, remain viable for about 8 years, creeping thistle, Cirsium irvens, for 20 years and field bindweed, Convolvulus arvensis, for about 50 years. Weed seeds have a tremendous capacity to disperse from one place to another through wind, water and animals including man. Many of times, weed seeds mimic with the crop seeds due to their size and get transported from one place to another along with them. Weeds compete with crops for water, soil, nutrients, light, and space, and thus reduce the crop yields. An estimate shows that weeds can deprive the crops 47% N, 42% P, 50% K, 39% Ca and 24% Mg of their nutrient uptake. 267-2243. Weeds are also act as alternate hosts that harbor insects, pests and diseases and other microorganisms. Alternate hosts of some of the pest and diseases crop post red gram caterpillar, alternate host Amaranthus, Datura. Custard rice hairy caterpillar Crotalaria sp wheat stem borer black rust echinocoloa, Panicum agropyron ribbons pearl millet maj ergot downy mildew concrus Ciliaris saccharum spontaneum. Weeds may have dual mode of reproduction. Most weeds are angiosperms and reproduce by seeds and vegetatively too. Weeds have environmental plasticity. Many weeds are capable of tolerating and growing under a wide range of climatic and edaphic conditions. Weeds reduce the quality of marketable agricultural produce. Contamination of weed seeds of Datura, Ajmone, Brassica etc. is harmful to human health, and weed seeds present in the produce cause odd odors sometimes. Weeds not only reduce yield but also interfere with agricultural operations. Weeds make mechanical sowing a difficult process and render harvesting difficult, leading to increased expenditure on labor, equipment and chemicals for their removal. Roots and other vegetative parts of perennial weeds are vigorous with large food reserves, enabling them to withstand environmental stress. Many weeds have adaptation mechanism that repels grazing, such as spines, or taste or odor. Some weeds release into the soil inhibitors of poisonous substances that may be harmful to the crop plants, human beings and livestock. E.g. hay fever and asthma, pollen of ambrosia and franceria. Dermatitis spartanium. 268. 2244. 2. Classification of weeds. 1. Based on lifespan. Based on lifespan, ontogeny, weeds are classified as annual weeds, biennial weeds and perennial weeds. Annual weeds, weeds that live only for a season or a year and complete their life cycle in that season or year, are called as annual weeds. These are small herbs with shallow roots and weak stems, produces seeds in profusion, and the mode of propagation is commonly through seeds. After seeding, the annuals die away and the seeds germinate and start the next generation in the next season or year following. Most common field weeds are annuals. The examples are monsoon annuals, Camelina bengalensis, Boerhaavia erecta. B. Winter annuals, Chenopodium album. B. Biennials, they complete the vegetative growth in the first season, flower and set seeds in the succeeding season and then dies. These are found mainly in non-cropped areas, e.g. Alternanthera agenta, Docus carota. C. Perennials. Perennials live for more than 2 years and may live almost indefinitely. They adapt to withstand adverse environmental conditions. They propagate not only through seeds but also by underground stems, roots, rhizomes, tubers etc. E.g. creeping perennials, they reproduce through seeds as well as with one of the followings: a rhizome, plants having underground stem, Sorghum halepens. B. Stolon, plants having horizontal creeping stem above the ground, Synodon dactylon. C. Roots, plants having enlarged root system with numerous buds, Convolvulus arvensis. D. Tubers, plants having modified rhizomes adapted for storage of food, Cyperus rotundus. 2. Based on ecological affinities. A. Wetland weeds, they are tender annuals with semi-aquatic habit. They can thrive well under waterlogged and in partially dry conditions. Propagation is chiefly by seed, e.g. Ammania baxifera, Eclipta alba B garden land weeds, irrigated lands. These weeds neither require large quantities of water like wetland weeds nor can they successfully withstand extreme drought as dryland weeds, e.g. Trianthema portulacastrum, Digera urvensis. 269. 2244. 2. Classification of weeds. 1. Based on lifespan. Based on lifespan, ontogeny, weeds are classified as annual weeds, biennial weeds and perennial weeds. Annual weeds, weeds that live only for a season or a year and complete their life cycle in that season or year, are called as annual weeds. These are small herbs with shallow roots and weak stems, produces seeds in profusion, and the mode of propagation is commonly through seeds. After seeding, the annuals die away and the seeds germinate and start the next generation in the next season or year following. Most common field weeds are annuals. The examples are monsoon annuals, Camelina bengalensis, Boehevia erecta B winter annuals, Canopodium album B biennials. They complete the vegetative growth in the first season, flower and set seeds in the succeeding season and then dies. These are found mainly in non-cropped areas, e.g. Alternanthera agenta, Docus carota. C. Perennials. Perennials live for more than 2 years and may live almost indefinitely. They adapt to withstand adverse environmental conditions. They propagate not only through seeds but also by underground stems, roots, rhizomes, tubers, etc. E.g. creeping perennials. They reproduce through seeds as well as with one of the followings: a rhizome, plants having underground stem, Sorghum halepens B stolon, plants having horizontal creeping stem above the ground, Synodon dactylon, C. roots, plants having enlarged root system with numerous buds, Convolvulus urvensis D. tubers, plants having modified rhizomes adapted for storage of food, Cyperus rotundus 2. Based on ecological affinities, wetland weeds. They are tender annuals with semi-aquatic habit. They can thrive well under waterlogged and in partially dry conditions. Propagation is chiefly by seed, e.g. Ammania baxifera, Eclipta alba B garden land weeds, irrigated lands. These weeds neither require large quantities of water like wetland weeds nor can they successfully withstand extreme drought as dryland weeds, e.g. Trianthema portulacastrum, Digera urvensis. 269. 2245. C. Dry lands weeds. These are usually hardy plants with deep root system. They are adapted to withstand drought on account of mucilaginous nature of the stem and hairiness. Tribulus terrestris, Argemone mexicana 3. Based on origin, indigenous weeds. All the native weeds of the country come under this group and most of the weeds are indigenous. Acalifa indica, Abutilon indicum, B. Introduced or exotic weeds. These are the weeds introduced from other countries. These weeds are normally troublesome and control becomes difficult. Parthenium hysterophorus, Phalaris minor, Acanthospermum hispidum 4. Based on cotyledon number. Based on number of cotyledones, they can be classified as dicots and monocots. Monocots Panicum flavidum, Echinochlua colonna, B. Dicots Crotalaria verrucosa, Indigofera viscosa V. Based on morphology. Based on the morphology of the plant, the weeds are also classified into 3 categories. This is the most widely used classification by the weed scientists. Grasses. All the weeds which come under the family Poaceae are called as grasses, which are characteristically having long narrow spiny leaves. The examples are Echinochlua colonum, Synodon dactylon. B. Sedges. The weeds belonging to the family Cyperaceae come under this group. The leaves are mostly from the base having modified stem with or without tubers. The examples are Cyperus rotundus, Fimbristyles meliaceae. C. Broad-leaved weeds. This is the major group of weeds. All dicotyledon weeds are broad-leaved weeds. The examples are Flavaria australasica, Digera urvensis, 3. Darch procumbens 6. Parasitic weeds. The parasite weeds are either total or partial, which means the weeds that depend completely on the host plant are termed as total parasites, while the weeds that partially depend on host plant for minerals and capable of preparing its food from the green leaves are called as partial parasites. Those parasites which attack roots are termed as root parasites, and those which attack shoot of other plants are called as stem parasites. 270. The typical examples are total root parasite Orband simua on tobacco, partial root parasite Striga lutea on sugarcane and sorghum, total stem parasite Cuscuta chinensis on lucerne and onion, partial stem parasite Cassitha filiformis on orange trees and Loranthus longiflorus on mango and other trees. 7. Poisonous weeds. The poisonous weeds cause ailment to livestock, resulting in death and cause great loss. These weeds are harvested along with fodder or grass and fed to cattle, or while grazing the cattle consume these poisonous plants, e.g. Datura fastosa, D. stramonium and D. metal are poisonous to animals and human beings. The berries of Withania somnifera and seeds of Abrus precatorius are poisonous. 271. 2245. Why? 3. Propagation and dissemination of weed seeds. Propagation is the process of multiplying or increasing the number of plants of the same species and at the same time perpetuating their desirable characteristics. There are 2 general methods of plant propagation: sexual and asexual propagation. Reproduction by seed. Reproduction by seed is called sexual reproduction. It requires pollination and fertilization of an egg, which results in seed that is capable of producing a new plant. Seed production varies greatly among and within weed species, in part due to environmental variability between years, competition from neighboring plants, and genetic variability. For example, while Canada thistle has been observed to produce as few as 680 seeds per plant, curly dock often produces more than 30,000 seeds per plant. Vegetative reproduction. In vegetative, asexual reproduction, a new plant develops from a vegetative organ such as a stem, root or leaf. Several modifications of these organs are common in perennial weeds, such as underground stems, rhizomes, above-ground stems, stolons, bulbs, combs, and tubers. Although vegetative structures generally do not survive as long in the soil as do seeds, very small structures can result in a new plant. Canada thistle, for example, can produce a new plant from as small as a 1/4-inch section of root. Vegetative reproduction can be as prolific as seed production. Yellow nutsedge, Cyperus esculentus, has been reported to produce more than 1,900 new plants and more than 6,800 tubers in 1 year. Some of the vegetative propagules are described as follows: rhizome, a horizontal, underground stem which can produce adventitious roots and shoots, i.e., new plants, at the nodes. A rhizome can be distinguished from a root because rhizomes have nodes, internodes, and scale leaves, rudimentary leaves. Roots do not have nodes or leaves. Johnson grass is example. Stolon, an above-ground stem that grows flat on the ground and can produce adventitious roots and shoots, i.e., new plants, at the nodes. Bermuda grass is example. Tuber, enlarged terminal portion of rhizomes, possess extensive storage tissues and axillary buds. Yellow nutsedge is example. Bulb, specialized underground storage organ consisting of fleshy leaves with a short stem at the base. Food storage in the leaves. Wild garlic is example. Creeping roots, horizontal roots modified for food storage and vegetative reproduction, can give rise to shoots, often deep in the soil. Carolina horse nettle is example. 272. 2245. Dissemination dispersal of weeds. Weeds are widely dispersed through various adaptations. This is one of the reasons why weeds survive and are found in almost all cultivated fields. Weed dispersal is the process by which seeds of weeds, fruits and propagules are carried from one place to another through one agent or the other. Dispersal of seed occurs in 4 dimensions: width 1, length 2, width, land habitat, soil surface area. Phenomena 3, height, soil depth, in the air. 4, time, shatters immediately after ripening, or need harvesting activity to release seed. Common weed dispersal agents. Wind. Many seeds are well adapted to wind travel. Cottony coverings and parachute-like structures allow seeds to float with the wind. Examples of wind-dispersed seeds include common milkweed, Asalpias syriaca, common dandelion, Canada thistle, and perennial softhistle, Suncus iovensis. Weed seeds and fruits that disseminate through wind possess special organs to keep them afloat. Such organs are papoose. It is a parachute-like modification of persistent calyx into hairs, e.g. Asteraceae family weeds. 3. Dark Procumbanes camose. Some weed seeds are covered with hairs, partially or fully, e.g. Calotropis sp. Feathery, persistent styles. Styles are persistent and feathery, e.g. Anemone sp. Balloon-modified papery calyx that encloses the fruits loosely along with entrapped air, e.g. Physalis minima. Wings. One or more appendages that act as wings, e.g. acer. Macrophyllum. Factors that influence wind dispersal: seed weight, seed shape, structures, wings or papoose, height of release, wind speed and turbulence. B. Water. Aquatic weeds disperse largely through water. They may drift either as whole plants, plant fragments, or as seeds with the water currents. Terrestrial weed seeds also disperse through irrigation and drainage water. Weed seed often moves with surface water runoff into irrigation water and ponds, where it is carried to other fields. Weeds growing in ditch banks along irrigation canals and ponds are the major source of weed seed contamination of irrigation water. Weed seed often remains viable in water for several years, creating a floating seed bank and allowing weeds to disperse over large areas in moving water. Field bindweed seed, for example, 273-2246, remains over 50% viable after being submerged in water for more than 4 years. Some seeds have special adaptations that aid in water travel. The seed pod of curly dog, for example, is equipped with pontoons that carry the floating seed. C. Animals. Several weed species produce seeds with barbs, hooks, spines, and rasps that cling to the fur of animals or to clothing and then can be dispersed to long distances. Farm animals carry weed seeds and fruits on their skin, hair, and hooves. This is aided by special appendages such as hooks, Xanthium strumarium, stiff hairs, Cencrus spp, sharp spines, Tribulus terrestris, and scarious bracts, Acharanthus aspera. Even ants carry a huge number of weed seeds. Donkeys eat Prosopis julifera pods. Weed seeds are often ingested and passed through the digestive tracts of animals. Animal droppings provide an ideal nutrient and moisture environment for weed germination. Only a small percentage of the seed remains viable after exposure to an animal's digestive enzymes. The ingested weed seeds are passed in viable form with animal excreta, which is dropped wherever the animal moves. This mechanism of weed dispersal is called endozoochory, e.g. lantana seeds by birds, Loranthus seeds stick on beaks of birds. D. Dispersal by man. Man disperses numerous weed seeds and fruits with raw agricultural produce. Weeds mature at the same time and height along with crop. Due to their similar size and shape as that of crop seed, man unknowingly harvests the weeds also, and aids in dispersal of weed seeds. Such weeds are called satellite weeds, e.g. Avena pachua, Phalaris minor. E. Dispersal by machinery. Weed seeds often are dispersed by tillage and harvesting equipment. Seeds move from field to field on the soil that sticks to tractor tires, and vegetative structures often travel on tillage and cultivation equipment and later dropping them in other fields to start new infestation. Disc-type cultivation equipment is less likely to drag vegetative plant parts than are shovels or sweeps. F. Intercontinental movement of weeds. Introduction of weeds from one continent to another is through crop seed, feedstock, packing material and nursery stock, e.g. Parthenium hysterophorus. G. Crop mimicry dispersal. Weed seed adaptations to look like crop seed, plant body or seed same size, shape, and morphology as crop, e.g. barnyard grass biotype looking like rice escapes hand weeding and is dispersed with rice. Nightshade fruit, berries, are same size, shape as dry beers, harvested and dispersed with beans. 274. 2246. H. As admixtures with crop shed, animal feed, hay, and straw. Weeds probably are spread more commonly during the seeding of a new crop or in animal feed and bedding than by any other method. Seed labels often indicate a tiny percentage of weed seed, but consider this example. If a legume seed contains 0.001% dodder, a parasitic annual, Cuscuta campestris, seed by weight, there will be 8 dodder seeds per 2 kilograms of legume seed. If the legume seed is sown in a field despite an extremely low dodder seed percentage by weight, the small size of the seed, combined with rapid early season growth, could result in an infested legume field within a single season. Persistence of weeds, adaptation. Persistence is an adaptive potential of a weed that enables it to grow in any environment. In an agricultural situation, the cropping system with its associated habitat management practices determines the persistence of weed species. It is largely influenced by climatic, edaphic, soil, and biotic factors, which affect its occurrence, abundance, range and distribution. Factors affecting persistence. A. Climatic factors. Climate can affect variations in cuticle development, pubescence, vegetative growth, vigor, competitiveness, etc. Climate thus has a profound effect on the persistence of weeds, which can adapt to a wide variety of climates. The important climatic factors are light, temperature, rainfall, wind and humidity. Light intensity, quality and duration are important in influencing the germination, growth, reproduction and distribution of weeds. Photoperiod governs flowering time, seed setting and maturation and on the evolution of various ecotypes within a weed species. Tolerance to shading is a major adaptation that enables weeds to persist. 2. Temperature. Temperature of atmosphere and soil affects the latitudinal and longitudinal distribution of weeds. Soil temperature affects seed germination and dormancy, which is a major survival mechanism of weeds. 3. Rainfall. Rainfall has a significant effect on weed persistence and distribution. More rainfall or less rainfall determines reproduction and survival. 4. Wind. Wind is a principal factor in the dissemination of weeds. B. Soil factors. Soil factors are soil water, aeration, temperature, pH and fertility level and cropping system. Some weed species are characteristically alkali plants, known as basophilic, pH 8.5, which can grow well in alkali soils, and those grow in acidic soil is known as acidophils. 275. 2246, Cade. Ed and D. Bu alkali grass Puccinelia spp Synodon dactylon common weeds Quack grass Agrophyron ripens Digitaria sanguinalis. Several weed species of Compositae family grow well in saline soils. A shift in soil pH towards acid side due to continuous use of ammonium sulfate as a N source could cause a shift in the weed spectrum. Many weeds can grow well in soils of low fertility level, however, can adapt well to soils of high fertility also. Weeds also has adaptation to moist soil, drought condition, etc. See biotic factors. In a cropping situation, the major effects on weeds are those exerted by the crop as it competes for available resources. Once a particular weed species is introduced, its persistence is determined by the degree of competition offered by the crop, and also the agricultural practices associated with the growing of a crop may encourage or discourage specific weeds, e.g. ponding of water, Synodon dyes, repeated cultivation discourage nut sedge. Crops that serve as hosts to parasitic weeds, sorghum striga sp, crop-induced stimulants are examples of other biotic factors. 276. 2247. 4. Crop weed competition and association of weeds with crop plants. Crop weed association. Weeds possess many growth characteristics and adaptations which enable them to successfully exploit the numerous ecological niches left unoccupied by crop cultures. Weeds compete with themselves and with crop plant. Among the more important adaptations relevant to competitive advantage are properly synchronized germination, rapid establishment and growth of seedlings, tolerance to shading effects by the crop or by other weeds at the time of establishment, quick response to available soil moisture and nutrients, adaptation to the most severe climatic situations of the habitat, adaptations to the edaphic regime, relative immunity to post-seeding soil disturbance, practices and resistance to herbicides that are used. In the initial stages of invasion by weeds of exposed ecological niches, only a very limited competition for resources by the crop and weed may occur, but as establishment of the crop-weed association is completed, competition for the available resources is more obvious. Association of weeds with a particular crop. Some important examples. Wheat Phalaris minor, Canopodium album, Avena pachua, Parthenium hysterophorus, Convolvulus arvensis, Cichorium intybus. Rice, Echinocloa colonna, Echinocloa cruzgalli, Cyperus spp, Altemanthera sp. Camelina bengalensis, Eclipta alba. Soya bean, Echinocloa colonna, Cyperus rotundas, Trianhema spp, Canopodium album, Convolvulus arvensis, Synodon dactylon. Chickpea, Canopodium album, Avena pachua, Arrobranch Phalaris minor, Cichorium intybus. Match, Echinocloa colonna, Cyperus spp Euphorbia hirta. Association of weeds with a particular crop is due to the following factors. 1. Morphological similarities. Morphological similar weeds are difficult to control both by mechanical and chemical methods. For example, plants of Echinocloa crusculi which resembles with that of rice are often uprooted with rice nursery, and well-grown plants of this weed transplanted with rice are not controlled with any herbicide recommended in the crop. 277. 2247. 2. Seed shading behavior. Majority of wheat plants complete their life cycle before the harvest maturity of the crops in which they are associated. The seeds which are shed in the field during one season become the source of infestation during the coming years. About 90 to 95% seeds of problematic weeds such as Phalaris minor, Avena ludoviciana, Echinocloa cruscul ischamum rugosum and Trianthema portulacastrum are shed in the field. This seed shading behavior is one of the main reasons of association of a particular weed with a specific crop when sown on same field regularly. 3. Escape removal through sieving. The seeds of few weeds are of the same size as of the crop grains seeds, and these cannot be separated out even by thorough sieving. These seeds are sown in the field along with crop seeds. The size of few weed seeds like Convolvulus arvensis, Rumex spinosus, Avena ludoviciana etc. are of the same size as of wheat grain, and these cannot be removed even with thorough sieving. The crop infested with these weeds should not be kept for seed production purposes. For congenial environment, the association of weeds not only depends upon crop but it is also influenced by cropping sequence. For example, field having infestation of wild oats in wheat crop can be eliminated if brought under rice, wheat rotation at least for 2 to 3 years. The wild oat seeds being porous absorb water and lose viability with continuous standing water. The continuous wet conditions eliminate wildcats but may give favor to Phalaris establishment due to congenial environmental conditions created with the cultivation of rice. 5. Germination in flushes. The weed seeds which have the tendency to germinate in many flushes are very difficult to control both with mechanical or chemical methods. The flush of weeds which appear after the adoption of weed control measures produce seed, and such type of weeds are rather permanently associated with the crop in spite of adoption of best weed management practices. For example, Phalaris minor seeds germinate in many flushes in wheat crop, and due to this character it is very difficult to control this weed. 6. Continuous use of one group of herbicides. With the continuous use of single group of herbicides over years leads to resistance phenomenon. For example, with continuous use of isoproteron in wheat, Phalaris minor has developed resistance to isoproteron in Punjab and Haryana. In spite of use of isoproteron even at higher doses, Phalaris minor was not killed. So this weed continued to associate wheat through physiological modification. Rotational use of herbicides helps a lot to avoid association of a particular weed with a particular crop. 7. Allelopathic effects. There are few weeds which are only associated with a particular host. In the absence of host plants, these weed seeds show no germination even when the seeds are in viable conditions. For example, Striga densiflora is a parasitic weed of sugarcane, and Arrobranch aegyptica 278. 2247 is of mustard. These host plants, crops, excrete some chemicals in soil, and these chemicals acts as stimulants for the germination of seeds of these parasitic weed and are associated with their specific host crops, while these do not germinate when other crops are sown in their habitat. Shifting of weed flora. A weed shift may be defined as the change in the composition or relative frequencies of weeds in a weed population, all individuals of a single species in a defined area, or community, all plant populations in a defined area, in response to natural or human-made environmental changes in an agricultural system. Weed shifts occur when weed management practices do not control an entire weed community or population. The management practice could be herbicide use or any other practice such as tillage, manure application, or harvest schedule that brings about a change in weed species composition. Some species or biotypes are killed by, or susceptible to, the weed management practice, others are not affected by the management practice, tolerant or resistant, and still others do not encounter the management practice, dormant at application. Those species that are not controlled can grow, reproduce, and increase in the community, resulting in a weed shift. Any cultural, physiological, biological, or chemical practice that modifies the growing environment without controlling all species equally can result in a weed shift. In the case of chemical weed control, no single herbicide controls all weeds, as weeds differ in their susceptibility to an herbicide. Susceptible weeds are largely eliminated over time with continued use of the same herbicide. This allows inherently tolerant weed species to remain, which often thrive and proliferate with the reduced competition. As a result, there is a gradual shift to tolerant weed species when practices are continuously used that are not effective against those species. A weed shift does not necessarily have to be a shift to a different species. For example, with a foliar herbicide without residual activity like glyphosate, there could also be a shift within a weed species to a late emerging biotype that emerges after application. For example, due to growing rice under alternating flooding regimes and residual soil moisture conditions prevalent in the Cauvery Delta region of Tamil Nadu, red sprangletop, Leptochlora chinensis, and European water clover, Marsilia quadrifolia, became predominant in rice fields by replacing barnyard grass, Echinocloa sp. In the eastern Indo-Gangetic plains, adoption of zero tillage has resulted in an increase in population of globally significant perennial weeds such as purple nut sedge, Cyperus rotundus, and Bermuda grass, Synodon dactylon. 279. 2252. Crop weed competition. Competition is struggle between two or more organisms for a limited resource, water, nutrient, light and space, that is essential for growth. Competition between crop plants and weeds is most severe when they have similar vegetative habit and common demand for available growth factors. Weeds appear much more adapted to agroecosystems than are crop plants. Without interference by man, weeds would easily wipe out the crop plants. Weeds compete with the crop for following growth factors. 1. Competition for nutrients. Weeds usually absorb mineral nutrients faster than many crop plants and accumulate them in their tissues in relatively larger amounts. Amaranthus sp. accumulates over 3% N on dry weight basis and is termed as nitrophil. Acherontes aspera, P. accumulates over 1.5% P205 Chenopodium sp and Portulaca sp. K. lovers with over 13% K20 in dry matter times. The associated weed is responsive to nitrogen and it utilizes more of the applied N than the crop. E.g. the N uptake by Echinocloa crusculi is more than rice. Nutrient removal by weeds leads to huge loss of nutrients in each crop season, which is often twice that of crop plants. For instance, at early stages of match cultivation, the weeds found to remove 9 times more of N, 10 times more of P and 7 times more of K. 2. Competition for moisture. In general, for producing equal amounts of dry matter, weeds transpire more water than do most of our crop plants. It becomes increasingly critical with increasing soil moisture stress, as found in arid and semi-arid areas. As a rule, C4 plants utilize water more efficiently, resulting in more biomass per unit of water. Synodon had almost twice as high transpiration rate as pearl millet. In weedy fields, soil moisture may be exhausted by the time the crop reaches the fruiting stage, i.e., the peak consumptive use period of the crop, causing significant loss in crop yields. 3. Competition for light. It may commence very early in the crop season if a dense weed growth smothers the crop seedlings. It becomes important element of crop weed competition when moisture and nutrients are plentiful. In dry land agriculture, in years of normal rainfall, the crop weed competition is limited to nitrogen and light. 280. 20-52. Unlike competition for nutrients and moisture, once weeds shade a crop plant, increased light intensity cannot benefit it. 4. Competition for space. CO2. Crop weed competition for space is the requirement for CO2 and the competition may occur under extremely crowded plant community condition. A more efficient utilization of CO2 by C4 type weeds may contribute to their rapid growth over C3 type of crops. Critical period of weed competition. Critical period of weed competition is defined as the shortest time span during the crop growth when weeding results in highest economic returns. The critical period of crop weed competition is the period from the time of sowing up to which the crop is to be maintained in a weed-free environment to get the highest economical yield. The weed competition in crop field is invariably severe in early stages of crop than at later stages, generally in a crop of 100 days duration. The first 35 days after sowing should be maintained in a weed-free condition. There is no need to attempt for a weed-free condition throughout the life period of the crop, as it will entail unnecessary additional expenditure without proportionate increase in yield. Critical period of crop weed competition and losses in yield in some crops. Crop critical period, days, % reduction in grain yield. Transplanted rice 30 to 45, 15 to 45, wheat 30 to 45, 20 to 40, match 15 to 45, 15 to 30, 40 to 60, green gram and black 25 to 50 gram, 30 to 60, chickpea 40 to 60. Groundnut 15 to 60, 40 to 50, cotton sugarcane 30 to 100 and 20, 15 to 25, 40 to 50, 20 to 30. 281 kanagraj.bioscience@gmail 2257. Factors affecting the competitive ability of crops against weeds. 1. Period of weed growth. Weeds interfere with crops at any time they are present in the crop. Thus weeds that germinate along with crops are more competitive. Sugarcane takes about 1 month to complete its germination phase while weeds require very less time to complete their germination. By that time crop plants are usually smothered by the weeds completely. First a quarter, 1/3 of the growing period of many crops is critical period. However, in a situation where weeds germinate late, as in dry land wheat and sorghum, the late stage weeding is more useful than their early weeding. In general, for most of the annual crops, first 20 to 30 days weed-free period is very important. 2. Density of weeds. Increase in density of weeds and ultimately decrease in yield is a normal phenomenon. However, it is not linear as few weeds do not affect the yields so much as other weed does and hence, it is a sigmoidal relationship. 3. Crop density. Increase in plant population decreases weed growth and reduce competition until they are self-competitive. Crop density and rectangularity are very important in determining the quantum and quality of crop environment available for the growth of weeds. Wide row spacing with simultaneous high, intra-row crop plant population may induce dense weed growth. In this respect, square planting of crops in which there are equal row and plant spacing should be ideal in reducing intra-crop plant competition. In wheat, reduced row spacing from 20 to 15 cm reduced the dry matter yield of Lolium and Phalaris sp. by 11.8% and 18.3%, respectively. 4. Type of weeds species. The type of weeds that occur in a particular crop influences the competition. Occurrence of a particular species of weed greatly influences the competition between the crop and weed. For example, E. crusculi in rice, Setaria viridis in corn and Xanthium sp. in soybean affects the crop yield. Flavaria australsica offers more competition than the grasses. 5. Type of crop species and their varieties. Crops and their varieties differ in their competing ability with weeds, e.g., the decreasing order of weed competing ability is as barley, rice, wheat and oat. High tolerance of barley to competition from weeds is assigned to its ability to develop more roots that are extensive during initial 3 weeks growth period than the others. Fast canopy forming and tall crops suffer less from weed competition than the slow growing and short stature crops. Dwarf and semi-dwarf varieties of crops are usually more susceptible to competition from weeds than the tall varieties because they grow slowly at initial stage. 282. 2258. In addition, their short stature covers the weeds less effectively. When we compare the crop weed competition between two varieties of groundnut TMV2, bunch, and TMV.3, spreading, TMV2 incurred a loss of over 30% pod yield under uncontrolled weed crop competition while TMV3 lost only about 15% of its yield. The main reason is due to the spreading nature of TMV3, which smothered weeds. Longer duration cultivars of rice have been found more competitive to weeds than the short duration ones. 6 soil factors: soil type, soil fertility, soil moisture, and soil reaction influences the crop weed competition. Elevated soil fertility usually stimulates weeds more than the crop, reducing thus crop yields. Fertilizer application of weedy crop could increase crop yields to a much lower level than the yield increase obtained when a weed-free crop is applied with fertilizer. Weeds are adapted to grow well and compete with crops in both moisture stress and ample moisture conditions. Removal of an intense moisture stress may thus benefit crops more than the weeds, leading to increased yields. If the weeds were already present at the time of irrigation, they would grow so luxuriantly as to completely overpower the crops. If the crop is irrigated after it has grown 15 centimeters or more in a weed-free environment, irrigation could hasten closing in of crop rows, thus suppressing weeds. Abnormal soil reactions often aggravate weed competition. Specific weed species suited to different soil reactions exist with us. Our crops grow best only in a specified range of soil pH. 7. Climate, adverse weather condition e.g. drought, excessive rains, extremes of temperature, will favor weeds since most of our crop plants are susceptible to climatic stresses. It is further intensified when crop cultivation is stratified over marginal lands. All such stresses weaken crops' inherent capacity to fight weeds. 8. Time of germination. In general, when the time of germination of crop coincides with the emergence of first flush of weeds, it leads to intense crop weed interference. Sugarcane takes about 1 month to complete its germination phase, while weeds require very less time to complete its germination. Weed seeds germinate most readily from 1.25 centimeters of soil, and few weeds can germinate even from 15 centimeters depth. Therefore, planting method that dries the top 3 to 5 centimeters of soil rapidly enough to deny weed seeds opportunity to absorb moisture for their germination usually postpones weed emergence until the first irrigation. By this time the crop plants are well established to compete with late germinating weeds. 283. Bioscience at E2258. 9. Cropping practices. Cropping practices, such as method of planting crops, crop density and geometry, and crop species and varieties have pronounced effects on crop weed interference. 10. Crop maturity. Maturity of the crop is yet another factor which affects competition between weeds and crop. As the age of the crop increases, the competition for weeds decreases due to its good establishment. Timely weeding in the early growth stages of the crop enhances the yield significantly. Losses caused by weeds. 1. Reduction in crop yield. Weeds compete with crop plants for nutrients, soil moisture, space, and sunlight. In general, an increase in 1 kg weed growth corresponds to reduction in 1 kg of crop growth. Depending on type of weed, intensity of infestation, period of infestation, the ability of crop to compete, and climatic conditions, the loss varies. Among the pests, weeds account for 45% reduction in yield, while the insects 30%, diseases 20%, and other pests 5%. 2. Loss in crop quality. If a crop contains weed seeds, it is to be rejected, especially when the crop is grown for seed. For example, the wild oat weed seeds are similar in size and shape of the crops like barley, wheat, and its admixture may lead to rejection for seed purpose. Contamination by poisonous weed seeds is unacceptable and increases costs of crop cleaning. The leafy vegetables much suffers due to weed problem, as the leafy weed mixture spoil the economic value. 3. Weeds as reservoirs of pests and diseases. Weeds form a part of community of organisms in a given area. Consequently, they are food sources for some animals, and are themselves susceptible to many pests and diseases. However, because of their close association with crop, they may serve as important reservoirs or alternate host of pests and diseases. 4. Interference in crop handling. Some weeds can make the operation of agricultural machinery more difficult, more costly, and even impossible. Heavy infestation of cyanidactylon causes poor plowing performance. Kanagraj, Faculty, Agriculture. 284. 2258. 5. Reduction in land value. Heavy infestation by perennial weeds could make the land unsuitable or less suitable for cultivation, resulting in loss in its monetary value. Thousands of hectares of cultivable area in rice-growing regions of India have been abandoned or not being regularly cultivated due to severe infestation of nutgrass, Cyperus rotundus, and other perennial grasses. 6. Limitation of crop choice. When certain weeds show heavy infestation, it will limit the growth of a particular crop. The high infestation of parasitic weeds such as Striga lutea may limit the growing of sorghum or sugarcane. 7. Loss of human efficiency. Weeds reduce human efficiency through physical discomfort caused by allergies, and poisoning weeds such as congress grass, Partenium hysterophorus, causes itching. Thorny weeds like Solanum spp restrict movement of farm workers in carrying out farm practices such as fertilizer application, insect and disease control measures, irrigation, harvesting, etc. B. Problems due to aquatic weeds. The aquatic weeds that grow along the irrigation canals, channels and streams restricts the flow of water. Weed obstruction causes reduction in velocity of flow and increases stagnation of water and may lead to high siltation and reduced carrying capacity. Aquatic weeds form breeding grounds for obnoxious insects like mosquitoes. They reduce recreational value by interfering with fishing, swimming, boating, hunting and navigation on streams and canals. 9 other problems. Weeds are troublesome not only in crop plants but also in playgrounds and roadsides etc. Alternanthera acinta and Tribulus terrestris occurs in many playgrounds causing annoyance to players and spectators. Allelopathy. Allelopathy is the detrimental effects of chemicals or exudates produced by one living plant species on the germination, growth or development of another plant species, or even microorganisms, sharing the same habitat. Allelochemicals are produced by plants as end products, byproducts and metabolites liberalized from the plants. They belong to phenolic acids, flavonoids, and other aromatic compounds with terpenoids, steroids, alkaloids and organic cyanides. Allelopathic effect of weeds on crops. 1. Mulch times leaves and inflorescence of Parthenium sp. Affect the germination and seedling growth. 285 Kanagraj 2259. Tubers of Cyperus esculentus affect the dry matter production. 2. Sorghum stem of Solanum affects germination and seedling growth times leaves and inflorescence of Parthenium affect germination and seedling growth. 3. Wheat times seeds of wild oat affect germination and early seedling growth. Tubers of C. rotundus affect dry matter production. Green and dried leaves of Argemone mexicana affect germination and seedling growth. Allelopathic effect of crop plants on weeds. 1. Root exudation of MAG inhibits the growth of Chenopodium album. 2. The cold water extracts of wheat straw when applied to weeds reduce germination and growth of Abutilon sp. Allelopathic effect of weeds on weeds. Inhibitors secreted by decaying rhizomes of sorghum halepens affect the growth of Digitaria sanguinalis and Amaranthus sp. Factors influencing allelopathy. A. Plant factors. I. Plant density. Higher the crop density, the lesser will be the allelochemicals it encounters. 2. Life cycle. If weed emerges later, there will be less problem of allelochemicals. 3. Plant age. The release of allelochemicals occurs only at critical stage. For egg in case of Parthenium, allelopathy occurs during its rosette and flowering stage. 4. Plant habit. The allelopathic interference is higher in perennial weeds. B. Plant habitat. Cultivated soil has higher values of allelopathy than uncultivated soil. B. Climatic factors. The soil and air temperature as well as soil moisture influence the allelochemicals potential. C. Soil factors. Physicochemical and biological properties influence the presence of allelochemicals. D. Stress factors. Abiotic and biotic stresses may also influence the activity of allelochemicals. Mechanism of action of allelochemicals times interfere with cell elongation. Interfere with photosynthesis times interfere with respiration. Interfere with mineral ion uptake. Interfere with protein and nucleic acid metabolism. 286. 2259. Use of allelopathy in biological control of weeds. 1. Use of cover crop for biological control. 2. Use of allelopathic chemicals as bioherbicides. Limitations of using allelopathic effect. There are many limitations in using allelopathic potentiality as a weed management tool. 1. Various abiotic and biotic factors, such as plant age, temperature, light and soil conditions, microflora, nutritional status and herbicide treatments influence the production and release of allelochemicals, although allelopathy is considered as a genetically influenced factor. 2. While moving in the soil, allelochemicals may undergo transformation as various factors regarding soil environment like physical, chemical, biological and physicochemical properties of soil may influence the activity of allelochemicals. After entry into soil, allelochemicals may be toxified or detoxified by microbes. 3. Many allelochemicals are very much expensive to synthesize in spite of having excellent herbicidal properties as for example, Tane toxin. 4. Some allelochemicals are toxic to human beings and are carcinogenic, e.g. Al toxin and fumonicin are toxic to mammalian cells. Sogoleone, for example, is reported to cause dermatitis. 5. The concentration of some inorganic ions affect the activity of allelochemicals. Mn, Fe, L, and Psi oxides cause polymerization of phenolic compounds into humic acids. However, Mn oxide is the most powerful catalyst. 287. N1232. 5. Integrated weed management, IWM. An integrated weed management may be defined as the combination of 2 or more weed control methods at low input levels to reduce weed competition in a given cropping system below the economical threshold level. Integrated weed management, IWM, approach aims at minimizing the residue problem in plant, soil, air and water. An IWM involves the utilization of a combination of mechanical, chemical and cultural practices of weed management in a planned sequence, so designed as not to affect the ecosystem. Why IWM? One method of weed control may be effective and economical in a situation and it may not be so in other situation. 2. No single herbicide is effective in controlling wide range of weed flora. 3. Continuous use of same herbicide creates resistance in escaped weed flora or causes shift in the flora. 4. Continuous use of only one practice may result in some undesirable effects, e.g. rice wheat. Cropping system, Fularis minor. 5. Only one method of weed control may lead to increase in population of particular weed. 6. Indiscriminate herbicide use and its effects on the environment and human health. Principles of integrated weed management. 1. IWM places the crop in competitive advantage over the weeds by manipulating the crop habitat by utilizing some biological differences between crops and weeds. 2. In IWM measures should be directed to reduce the survival mechanism of weeds in the soil. 3. Crop cultural practices should be incorporated to discourage the establishment of the perennial and parasitic weeds, e.g., crop rotation. 4. Any individual element of the weed management should be eco-friendly and it should not be harmful to the environment. 5. Weed management practices should be flexible to accommodate possible innovations and experiences of progressive farmers. Advantages of IWM. It shifts the crop weed competition in favor of crop times, prevents weed shift towards perennial nature times, prevents resistance in weeds to herbicides, no danger of herbicide residue in soil or plant, no environmental pollution, gives higher net return times, suitable for high cropping intensity. 288. 1233. Chemical herbicides, ingredient weed management, biological, ecological, cultural, methods of weed control. There are two broad methods of weed control, 1. Preventive methods, 2. Curative methods. Eradication. 2. Control measures. 1. Preventive method, prevention of introduction and spread of weeds in an entirely new locality is termed as preventive method. It is essential to know that how weeds disseminate. By taking following measures, weed spread can be prevented from entering into a new locality. 1. Sowing of weed-free clean seed. The seed contaminated with weed seed is a good source of spread of weeds. It becomes hard to separate the weed seed from the crop seed. For example, cruciferous crops like radish, cauliflower, cabbage, broccoli, etc. are well mixed with the seed of Satyanashi, Ajmone mexicana. Such impure seed should be discarded for sowing. 2. Use of clean implements. While operating agricultural implements like cultivator, harrow, and seed drill, etc. in weed-infested field, care must be taken that multiplication part of weed like rhizome, bulb, tubers, stem is not being carried along. The agricultural implements should be cleaned properly. Only then these should be used in other fields. This helps in controlling spread of the weeds. 3. Removal of weeds along canal and irrigation channel. Weed seed get transported through water and reach the field. Removal of weeds growing along the sides of canal or irrigation channel is necessary. 289. 1233. 4. Care in transplanting of seedling plantlets. Many horticultural plants like all transplanted vegetables, flowers, and fruits are transplanted in the field with soil attached to their root. Infestation of soil with weed may contaminate a new field. Use of well-rotten manure. Weed seeds have good viability. The seed of Hirankuri, Convolvulus arvensis remain viable for as long as 50 years. Dube, Synodon dactylon, and Matha, Cyperus rotundum seed viability lasts for 2 and 5 years, respectively. For making manure, the cow dung is generally heaped. If the heaping period is short, the seed do not lose its viability and grows in the field wherever manure is applied. So only well-rotten manure should be used. 6. Avoiding passing of cattle from weed-infested area. Grazing in weed-infested field followed by allowing passage of cattle in new field favors dissemination of weed seed. The weed seeds after passing through alimentary canal of the animal come out through dung, where it gives rise to weed. Some weed seeds also stick to the legs and skin of the animals and get transported to some other place where it germinate and grow as a weed. 7. Crop management practices. All such practices which favor the growth of main crop only disfavor the growth of weed. The following management practices have smothering effect on weed and must find place in cropland to prevent weed spread. Proper crop rotation prevents establishment of weeds. Higher plant population per unit area smothers the growth of weed. Proper placement of fertilizer in the root zone of the seed favors the growth of crop only. The weeds deprive of nutrients and their growth is restricted. Fast and vigorous growing varieties by virtue of their larger leaf canopy cause smothering effect on the growth of weed. Such crops should receive preference to prevent spread of the weed. 8. Enforcement of weed laws. In India, many noxious weeds grow in the fields and pose great economic and health hazard. Noxious weeds are those perennial weeds which are reproduced by seeds, stem, roots, and other reproductive parts as well and are very difficult to control. Partenium hysterophorus, Striga sp. Cyperus rotundus, Synodon dactylon etc. are noxious weeds that grow in many horticultural crops. In India, no weed laws are in force except in Karnataka where Partenium has been declared as a noxious weed. 9. Quarantine laws. Quarantine laws impose legal restrictions on the movement of the agricultural material. If there were adequate quarantine laws, the Partenium and Ajmone which widely grows in vegetable and flower field may not have entered India. Creating isolation between widely weed-infested area and new area is essential by enforcing and observing quarantine properly. 290. 1233. X. Use of pre-emergence herbicides. Herbicides which are used before the emergence of weeds either before or after planting of crop is a good preventive measure for preventing weed infestation. Such herbicides either inhibit seed germination or kill young seedlings before they get established. 2. Curative methods. 1. Eradication. Eradication means elimination of weeds after they have become established in an area and control methods are adopted where prevention and eradication have failed, so control measure as a rule eradicate the weeds and make it possible to raise the crops in spite of their presence. Constant control measures applied for several years help in practical elimination of weeds. Eradication is however impractical and if practiced in large scale it may be possible to eradicate certain weeds which are limiting in their spread. 2. Methods of weed control. Weed control methods are grouped into cultural, physical, chemical and biological. Every method of weed control has its own advantages and disadvantages. No single method is successful under all weed situations. Many a time, a combination of these methods gives effective and economic control than a single method. 1. Mechanical weed control. The mechanical methods include tillage, hoeing, hand weeding, digging, sickling, mowing, burning, flooding, mulching, etc. 1. Tillage. Tillage removes weeds from the soil resulting in their death. It may weaken plants through injury of root and stem pruning, reducing their competitiveness or regenerative capacity. Tillage also buries weeds. Tillage operation includes plowing, disking, harrowing and leveling. 2. Hoeing. Hoe is a very useful implement to control weeds. Hoeing is particularly more effective on annuals and biennials as weed growth can be completely destroyed. In case of perennials, it destroyed the top growth with little effect on underground plant parts resulting in regrowth. 3. Hand weeding. It is done by physical removal or pulling out of weeds by hand or removal by implements called kurfi, which resembles sickle. It is probably the oldest method of controlling weeds and it is still a practical and efficient method of eliminating weeds in cropped and non-cropped lands. 291. 1234. 4. Digging. Digging is very useful in the case of perennial weeds to remove the underground propagating parts of weeds from the deeper layer of the soil, e.g. Cyperus rotundus. 5. Sickling and mowing. Sickling is also done by hand with the help of sickle to remove the top growth of weeds. It is popular in sloppy areas where only the tall weed growth is sickled, leaving the root system to hold the sail in place to prevent soil erosion. Mowing is a machine-operated practice mostly done on roadsides and in lawns. 6. Burning. Burning or fire is often an economical and practical means of controlling weeds. It is used to a. Dispose of vegetation, b. Destroy dry tops of weeds that have matured, c. Kill green weed growth in situations where cultivations and other common methods are impracticable. 8. Flooding. Flooding is successful against weed species sensitive to longer periods of submergence in water. Flooding kills plants by reducing oxygen availability for plant growth. The success of flooding depends upon complete submergence of weeds for longer periods. Merits of mechanical method. 1. Oldest, effective and economical method. 2. Large area can be covered in shorter time. 3. Safe method for environment. 4. Does not involve any skill. 5. Weeding is possible in between plants. 6. Deep-rooted weeds can be controlled effectively. Demerits of mechanical method. 1. Labor-consuming. 2. Possibility of damaging crop. 3. Requires ideal and optimum specific condition. 2. Cultural weed control. Several cultural practices like tillage, planting, fertilizer application, irrigation, etc., are employed for creating favorable condition for the crop. These practices, if used properly, help in controlling weeds. In cultural methods, tillage, fertilizer application and irrigation are important. 292. 1234. 1. Field preparation. The field has to be kept weed-free. Flowering of weeds should not be allowed. This helps in prevention of build-up of weed seed population. 2. Summer tillage. The practice of summer tillage or off-season tillage is one of the effective cultural methods to check the growth of perennial weed population in crop cultivation. Initial tillage before cropping should encourage clod formation. These clods, which have the weed propagules upon drying, desiccate. The same subsequent tillage operations should break the clods into small units to further expose the shriveled weeds to the hot sun. 3. Maintenance of optimum plant population. Lack of adequate plant population is prone to heavy weed infestation. Therefore, practices like selection of proper seed, right method of sowing, adequate seed rate, protection of seed from soil-borne pests and diseases, etc. are very important to obtain proper and uniform crop stand capable of offering competition to the weeds. 4. Crop rotation. The possibility of a certain weed species or group of species occurring is greater if the same crop is grown year after year. The obnoxious weeds like Cyperus rotundus can be controlled effectively by including lowland rice in crop rotation. 5. Growing of intercrops. Intercropping suppresses weeds better than sole cropping and thus provides an opportunity to utilize crops themselves as tools of weed management. Many short-duration pulses with green gum and soybean effectively smother weeds without causing reduction in the yield of main crop. 6. Mulching. Mulch is a protective covering of material maintained on soil surface. Mulching has smothering effect on weed control by excluding light from the photosynthetic portions of a plant and thus inhibiting the top growth. It is very effective against annual weeds and some perennial weeds like Synodon dactylon. Mulching is done by dry or green crop residues, plastic sheet or polythene film. 7. Crop competition, simplest method of weed control. Weeds are strong competitors. They take the lion's share of the plant nutrients. For every 1 kg of weed growth, soil produces about 1 pound less of crop. Thick crops strongly compete for nutrients by weeds. Most common smother crops are sorghum, fodder, clovers, lucerne, soybean and sun hemps. Fodder crops are smothering as their frequent cuttings destroy the top growth of weeds before they set seeds. Smother crop weakens the underground parts of the weeds and they are easily killed by the cultivation that follows. 293. 1235. 8. Crop management practices. Good crop management practices that play an important role in weed control are: A. Vigorous and fast-growing crop varieties are better competitors with weeds. B. Proper placement of fertilizers ensures greater availability of nutrients to crop plants, thus keeping the weeds at a disadvantage. C. Better irrigation practices to have a good head start over the weeds. D. Proper crop rotation program. E. Higher plant population per unit area results in smothering effect on weed growth. Merits of cultural method. 1. Low cost for weed control. 2. Easy to adopt. 3. No residual problem. 4. Technical skill is not involved. 5. No damage to crops. 6. Effective weed control. 7. Crop weed ecosystem is maintained. Demerits of cultural method. 1. Immediate and quick weed control is not possible. 2. Weeds are kept under suppressed condition. 3. Perennial and problematic weeds cannot be controlled. 4. Practical difficulty in adoption. 3. Chemical methods. Herbicides are chemicals capable of killing or inhibiting the growth of plants. Many chemicals have shown high codes of selectivity to certain crops, killing the weeds effectively. But proper selection of the herbicide, its rate, time, and method of application are very important to obtain the desired degree of weed control and crop selectivity. For biological control, use of living organisms viz. Insects, disease organisms, herbivorous fish, snails or even competitive plants for the control of weeds is called biological control. In biological control method, it is not possible to eradicate weeds but weed population can be reduced. Qualities of bioagent. The bioagent must feed or affect only one host and not other useful plants. It must be free of predators or parasites. 294. 1235. It must readily adapt to environment conditions. The bioagent must be capable of seeking out itself to the host. It must be able to kill the weed or at least prevent its reproduction in some direct or indirect way. It must possess reproductive capacity sufficient to overtake the increase of its host species without too much delay. Merits 1. Least harm to the environment. 2. No residual effect. 3. Relatively cheaper and comparatively long-lasting effect. 4. Will not affect non-targeted plants and safer in usage. Demerits 1. Multiplication is costlier. 2. Control is very slow. 3. Success of control is very limited. 4. Very few host-specific bioagents are available at present. Outstanding and feasible examples of biological weed control. A. In India, prickly pear Opuntia sp is controlled by cochineal insects Dactylopios indicus, and D. tomentosus p. lantana camera is controlled by larvae of Crocidosma lantana. A moth bores into the flower stems, eat flowers and fruits. C. Cyperus rotundus, Baccara verutna. A moth borer. D. Herbivorous fish Tilapia controls algae, common carp. A non-herbivorous fish controls submersed aquatic weeds. E. The use of Mexican beetle, Zygogramma bicolorata, has shown great promise in management of Partenium. Bioherbicides. Mycoherbicides. The use of plant pathogens which are expected to kill the targeted weeds. These are native pathogen cultured artificially and sprayed just like post-emergence herbicides each season on target weed, particularly in crop areas. Devine, a liquid suspension of fungal spores of Phytophthora palmivora causes root rot for control of stranglevine, Morenia odorata, in citrus. Coligo vegetable powder containing fungal spores of Caletotrichum gliosporoides causes stem and leaf blight for control of joint wedge, Eschiomone virginica, in rice, soybean. 295. 1235. Bipolaris. A suspension of fungal spores of Polaris sorghaic for control of Johnsonga, sorghum hailpins. Biolophos, a microbial toxin produced as fermentation product of Streptomyces hygroscopicus for control of non-specific, general vegetation. Challenges of IWM. The current challenge for producers is to manage herbicides and other inputs in a manner that prevents adapted species from reaching troublesome proportions. Weed ecology. For farmers to completely benefit from integrated weed management technologies, mechanistic research must be conducted in weed ecology, genetics and physiology to increase basic understanding of the processes that regulate weed crop interactions, weed population dynamics, adaptation and persistence under various management practices. On-farm assessment of available IWM options. The IWM options identified by researchers must be tested in the farmers' fields to assess their effectiveness and economic viability. Despite decades of research and extension efforts in popularizing the integrated weed management (WM) practices, its importance and effectiveness are not completely understood and hence less adopted by the farmers. Need for knowledge-based decision-making tools. There is a need to develop a larger database of weed ecology and biology characteristics, develop, improve and refine integrated weed management system simulation models. The challenge for weed scientists is to develop innovative, effective, economical and environmentally safe IWM systems that can be integrated into current and future cropping systems to bring a more diverse and integrated approach to weed management. 296. 1236. 6. Chemical method of weed control using chemicals, generally referred to as herbicides, for the control of weeds is called chemical weed control. The discovery of 2,4-D sodium salt in 1944 is a landmark in herbicide usage. Merits. Herbicide can be recommended for adverse soil and climatic conditions, as manual weeding is highly impossible during monsoon season. Herbicide can control weeds even before they emerge from the soil so that crops can germinate and grow in completely weed-free environment at early stages. It is usually not possible with physical weed comfort. Weeds, which resemble like crop in vegetative phase, may escape in manual weeding. However, these weeds are controlled by herbicides. Herbicide is highly suitable for broadcasted and closely spaced crops. Controls the weeds without any injury to the root system of the associated standing crop, especially in plantation crops like tea and coffee. Reduces the need for pre-planting tillage. Herbicides control the weed in the field itself or in situ controlling, whereas mechanical method controls many perennial weed species may lead to dispersal of weed species through seed. It is profitable where labor is scarce and expensive. Suited for minimum tillage concept. Highly economical. Demerits. Pollutes the environment, affects the soil microbes if the dose exceeds. Herbicide causes drift effect to the adjoining field. It requires certain amount of minimum technical knowledge for calibration. Leaves residual effects. Some herbicide is highly costlier. Suitable herbicides are not available for mixed and intercropping system. Classification of herbicides. 1. Based on method of application. 1. Soil applied herbicides. Herbicide act through root and other underground parts of weeds. Egg fluchloralin. 2. Foliage applied herbicides. Herbicide primarily active on the plant foliage. Egg glyphosate, paraquat. 297. 1236. 2. Based on mode of action. 1. Selective herbicide. A herbicide is considered as selective when in a mixed growth of plant species. It kills some species without injuring the others. Atrazine. 2. Non-selective herbicide. It destroys majority of treated vegetation. G. Paraquat. 3. Based on mobility. I. Contact herbicide. A contact herbicide kills those plant parts with which it comes in direct contact, e.g. paraquat 2. Translocated herbicide. Systemic herbicide. Herbicide which tends to move from treated part to untreated areas through xylem-phloem tissues depending on the nature of its molecule. E.G. Glyphosate 4. Based on time of application. I. Pre-plant application. Application of herbicides before the crop is planted or sown. Soil application as well as foliar application is done here. For example, flucloraline can be applied to soil and incorporated before sowing rain-fed groundnut, while glyphosate can be applied on the foliage of perennial weeds like Cyperus rotundus before planting of any crop. 2. Pre-emergence. Application of herbicides before a crop or weed has emerged. In case of annual crops, application is done after the sowing of the crop but before the emergence of weeds, and this is referred as pre-emergence to the crop, while in the case perennial crops it can be said as pre-emergence to weeds. For example, soil application by spraying of atrazine on third DAT to sugarcane can be termed as pre-emergence to cane crop, while soil application by spraying the same immediately after a rain to control a new flush of weeds in an intercultivated orchard can be specified as pre-emergence to weed. E.G. Atrazine, pendimethalin, butachlor, thiobane carb, pretilachlor. 3. Post-emergence. Herbicide application after the emergence of crop or weed is referred as post-emergence application. When the weeds grow before the crop plants have emerged through the soil and are killed with a herbicide, then it is called as early post-emergence. For example, spraying 2,4-D salt to control parasitic weeds striga in sugarcane is called as post-emergence, while spraying of paraquat to control emerged weeds after 10 to 15 days after planting potato can be called as early post-emergence. E.G. Glyphosate, paraquat, 2,4-D salt. 298. 1237. 5. Based on molecular structure or chemical nature. Herbicides are primarily grouped into inorganic and organic herbicides. I. Inorganic compounds. Inorganic herbicides do not contain carbon atoms in their molecules. They were the first chemicals used for weed control. Arsenic acid, arsenous acid, sulfuric acid are acid type of inorganic herbicides, whereas sodium arsenate, sodium chlorate, copper sulfate etc. are inorganic salts too. Organic compounds. These herbicides contain carbon atoms in their molecules. They may be oils or non-oils, e.g., diesel oil, xylene type of aromatic oils. Majority of present-day herbicides are non-oils: amides, alchlor, butachlor, propchlor, metallachlor, diphenamide, propapyltriazenes, atrazine, simazine, phenoxy acids, 2,4-D, 2,4,5-T, MCPA, dinitronilins, flucloraline, trifluoralin, pendimethalin, nitralin, isoproterin. Herbicide formulation. Herbicides in their natural state may be solid, liquid, volatile, non-volatile, soluble or insoluble. Hence these have to be made in forms suitable and safe for their field use. And herbicide formulation is prepared by the manufacturer by blending the active ingredient with substances like solvents, inert carriers, surfactants, stickers, stabilizers, etc. Objectives in herbicide formulations are ease of handling, high controlled activity on the target plants. Need for preparing herbicide formulation to have a product with physical properties suitable for use in a variety of types of application equipment and conditions. To prepare a product which is effective and economically feasible to use. Times to prepare a product which is suitable for storage under local conditions. Types of formulation. I. Emulsifiable concentrates, e.g. a concentrated herbicide formulation containing organic solvent and adjuvants to facilitate emulsification with water, e.g. butachlor 2. Wettable powders, WP. A herbicide is absorbed by an inert carrier together with an added surface acting agent. The material is finely ground so that it may form a suspension when agitated with a required volume of water, e.g. atrazine. 299. 1237. 3. Granules. G. The inert material, carrier, is given a granular shape and the herbicide, active ingredient, is mixed with sand, clay, vermiculite, finely ground plant parts, ground corn cobs, as carrier material. Alachlor granules. 4. Water-soluble concentrates, WSC. Paraquat. Mode of action of herbicides. The term mode of action refers to the sequence of events from absorption into plants to plant death. The mode of action of the herbicide influences how the herbicide is applied. For example, contact herbicides that disrupt cell membranes such as aciflurfen, blaze, or paraquat, gramoxone extra, need to be applied post-emergence to leaf tissue in order to be effective. Seedling growth inhibitors, such as trifluoralin, treflan, and alachlor, lasso, need to be applied to the soil to effectively control newly germinated seedlings. To be effective, herbicides must 1. Adequately contact plants. 2. Be absorbed by plants. 3. Move within the plants to the site of action, without being deactivated, and 4. Reach toxic levels at the site of action. The application method used, whether pre-plant incorporated, pre-emergence, or post-emergence, determines whether the herbicide will contact germinating seedlings, roots, shoots, or leaves of plants. The herbicide families listed below are grouped on the basis their mode of action. 1. The growth regulator herbicides, 2,4-D, MCPP, dicamba, and triclopyr. These are mostly foliar applied herbicides which are systemic and translocate in both the xylem and phloem of the plant. They mimic natural plant auxins, causing abnormal growth and disruption of the conductive tissues of the plant. The injury from this family of herbicides consists of twisted, malformed leaves and stems. 2. The inhibitors of amino acid synthesis, glyphosate, halosulfuron, imidacetapyr, and sulfomethurone. Both foliar and soil applied herbicides are in this family. Glyphosate translocates in the phloem with photosyntat produced in the leaves. Others in this family move readily after root or foliar absorption. These herbicides inhibit certain enzymes critical to the production of amino acids. Amino acids are the building blocks of proteins. Once protein production stops, growth stops. Symptoms are stunting and symptoms associated with lack of critical proteins. 300. 1237. 3-cell membrane disruptors with soil activity, oxyfluorphen, lactofen, and acifluorphen, soil and foliar applied with limited movement in soil. These herbicides enter the plant through leaves, stems and roots, but are limited in their movement once they enter the plant. Membrane damage is due to lipid peroxidation. Symptoms are necrosis of leaves and stem. 4 lipid biosynthesis inhibitors, diclofop, fluidifop, cetoxidime, and clitodime. Foliar applies. Diclofop has both soil and foliar activity. Herbicides in this family move in both the xylem and phloem of the plant and inhibit enzymes critical in the production of lipids. Lipids are necessary to form plant membranes, which are essential to growth and metabolic processes. Symptoms include stunting and death of tissue within the growing points of plants. 5 pigment inhibitors, norfluoresin, fluoridin, and amitrole. Soil applied and move in the xylem except amitrole, which moves in both phloem and xylem. These herbicides inhibit carotenoid biosynthesis, leaving chlorophyll unprotected from photooxidation. This results in foliage which lacks color. Symptoms include albino or bleached appearance of foliage. 6 growth inhibitors of shoots, thiocarbamate herbicides including EPTC, cycloat, pebulate, and molinate. Soil applied and somewhat volatile, requiring incorporation. Enter the plant through the roots and translocated through the xylem with the transpiration stream to the growing points in the shoot. Mode of action is unclear, but affects developing leaves in growing points of susceptible plants. Symptoms include stunting and distortion of seedling leaves. 7 herbicides which disrupt cell division, trifluralin, DCPA, dithiopia, orazalin, pronamide, pendimethalin, and napropamide. All are soil applied, with limited movement in the soil. Absorbed through roots or emerging shoot tips. Once absorption takes place, movement is limited, site of action is near the site of absorption. These herbicides inhibit cell division or mitosis, except pronamide and napropamide which stop cell division before mitosis. Symptoms include stunting and swollen root tips. 8 cell membrane disruptors. No soil activity, paraquat, dicut, glufosinate, acids, oils, soaps. These herbicides are foliar applied with no soil activity. They enter the plant through the leaves and stems and do not move significantly within the plant once absorbed. These herbicides either act directly on cell membranes, acids, soaps, oils, or react with a plant process to form destructive compounds which result in membrane damage. Symptoms include rapid necrosis of the leaves and stem. 9 inhibitors of photosynthesis, atrazine, simazine, metribuzin, cyanazine, prometrine, diurone, linurone, tbuturone, and bromycil. These are soil-applied herbicides. However, all except simazine also have foliar activity. They move readily in the plant in the xylem with the transpiration stream. 301. 1. 1237. Where they concentrate in the leaves at the site of photosynthesis. Once there they block the electron transport system of photosynthesis, causing a buildup of destructive high-energy products which destroy chlorophyll and ultimately the leaf tissues. Symptoms include chlorotic, yellowed, leaves which become necrotic. Selectivity of herbicides and factors affecting it. It is the phenomenon mechanism by which in a mixed stand of plants, some plant species, eggweeds, are preferentially controlled or killed, while others, egg crop plants, remain unaffected or negligibly affected when a herbicide is applied to them. Selectivity is a complex phenomenon achieved by complex interaction between plant, herbicide and environment, climate and soil. Selective mechanism of herbicides may occur due to these aspects. 1. Differential rate of absorption of herbicides. 2. Differential rate of translocation of herbicides. 3. Differential rate of deactivation of applied herbicides. 4. Protoplasmic resistance to the specific herbicide. 1. Differential absorption of herbicides may occur due to many reasons. Plant species may differ in their morphology and growth habits. Herbicides may be applied at different times by different methods. Use of antidotes and adsorbents to prevent herbicide absorption by non-target plants. Herbicide formulations may differ in their ability to contact with non-target plants. 2. Differential rates of translocation of herbicides. Plants can translocate herbicide through the plant as much herbicide it absorbs. Equal amounts of herbicides may be absorbed by plants and weeds, but translocation rates may be different. Always faster translocation. Translocation does not mean quick killing. In certain cases it will help the plants in escaping specific herbicide action. 3. Differential rates of deactivation of herbicides. This deactivation may be a process of metabolism change in molecular structure of applied herbicides inside the plants. Reverse metabolism. This is an enzymatic beta oxidation process. Intermediate chemical compounds are more phytotoxic than original compounds, parent compounds. Conjugation coupling of intact herbicide molecule with some plant cell constituents in living ST plants. 302. 1238. 4. Differential protoplasmic resistance. Protoplasm of different plant species differing in withstanding athenormal deficiencies or X constituents that may be induced in the presence of some specific herbicide molecules. Buffering mechanism of protoplasm of plants is affected differently by different herbicides. Efficacy of herbicides and factors affecting it. Efficacy of herbicide is typically defined as a substantial reduction of the target plant that results in a diminishment of its negative effects on the system. Efficacy parameters. 1. Weed control efficiency. Percent. Weed control efficiency, WCE, denotes the magnitude of weed reduction due to weed control treatment. It is expressed in percentage. WCE is equal to dry weight of weeds in unweeded control, dry weight of treatment plot, dry weight of weeds in unweeded control x 100. 2. Weed index, percent. Weed index is defined as the magnitude yield reduction due to presence of weeds in comparison with weed-free check. In other words, weed index expresses the competition offered by weeds measured by percent reduction in yield owing to their presence in the field. Weed index was calculated by using following formulae. Weed index, percent, is equal to x 100 where x is equal to total yield from the weed-free check, y is equal to total yield from the treatment for which weed index has to be calculated. Factors affecting herbicide effectiveness. A. Plant factors. 1. Morphology of plants. 11. Physiology of plants. B. Herbicide factors. C. Environmental factors. 303. 1238. 1. Morphology of plants. I. Leaf foliage canopy. I. Nature and orientation of leaf. The nature and orientation of leaves directly affect the interception and retention of a herbicide and indirectly its subsequent penetration and translocation. Broadleaf plants having bigger size, greater area and horizontal orientation of leaves are likely to intercept more amount of herbicide than grasses which have narrow, erect and upright leaves of smaller size and area. As a result, broadleaf plants are more prone to be killed by a herbicide than grasses. B. Pubescence and waxiness of leaf canopy. A waxy surface or one that has a lot of hairs will reduce amount of herbicide contacting plant leaf surface. If hairs are less on the leaf surface, the droplets retained in between hairs may require more time to be absorbed. C. Composition and thickness of cuticle. The thickness of cuticle directly affects the penetration of a herbicide and indirectly its translocation through plant system. The thicker the cuticle, lesser is the penetration. D. Age stage of the plants. Generally younger are the plants, greater are the susceptibility. Younger plants usually have tender, less developed roots, more meristematic tissues and growing activities, elongate very rapidly and less thick cuticle than older ones and therefore, are likely to be more prone to herbicide. 2. Physiology of plants. I. Uptake absorption. The availability of herbicide may be same on the site of application, e.g., leaves, stems, roots, but uptake may vary due to genetic factors. Herbicide absorption by roots in most cases is passive and therefore, factors affecting transpiration may affect absorption of herbicide by roots. 2. Translocation. Differential absorption of herbicides by plants results differences in the translocation of herbicides inside plants. Susceptible plants translocate more amount of herbicide compared to resistant or tolerant ones. 3. Metabolism of herbicide. Plants differ in their ability to metabolize herbicides. Once a herbicide has entered into the plants, metabolism is faster than accumulation in some plants. As a result, lethal herbicide amount at the site of action hardly build up and plants show tolerance, whereas reverse is true for the plants showing susceptibility or mortality. For example, isoproterenone, a substituted phenyluric herbicide and photosynthesis, PS2, inhibitor, is selective to wheat due to its increased detoxification inside wheat plants, whereas grass weeds, namely Phalaris minor and Avena fatua, are killed because of the decomposition of herbicide is slower. 304. 1238. B. Herbicide factors. I. Chemical family. Herbicides are synthesized with chemical modification of a base compound and are grouped under a chemical family class named after the base compound, e.g. dinitrophenols, dinitronilins, etc. Herbicides of one chemical class differ from those of another class. Herbicides within a class differ from each other as well as may have some common physical and chemical properties, toxicities, absorption, water solubility, vapor pressure, which render them selective or non-selective to group of plants. 2. Formulation of herbicide. In granular formulation, the herbicide toxicant remains impregnated inside the granules and does not come in direct contact of crops and weeds when spread in the field. Granular formulation thus offers a general selectivity to established crop plants and associated germinating weeds. Granular ensures slow release and thereby lower leaching of the herbicide in soil, which provides greater opportunity of selective absorption of herbicide by crops and weeds emerging from different stratum of soil. Herbicides applied on the soil surface hardly reach to crop roots lying deeper in soil and thus crops remain safe. 3. Time of herbicide application. Pre-plant incorporation, PPI, and pre-emergence applications offer more selectivity to a crop than a post-emergence application. In their case, selectivity to crops is achieved mainly due to depth protection. However, plant morphology, physiology, metabolism, and age stage of plants play a dominant role towards selectivity under post-emergence application. 4. Use of adjuvants. The effect of a herbicide and non-toxic adjuvant applied in combination on a plant is said to have an enhancement effect if the response is greater than that obtained when the herbicide is used at the same rates without the adjuvant, e.g. mixing ammonium sulfate with glyphosate. See environmental factors, climatic factors. I. Temperature. If there is an increase in temperature, greater uptake and translocation of herbicides are expected to occur because of higher transpiration. Metabolism may also take place at higher rate. For example, isoproterenone and barbane have faster metabolism at higher temperatures and thereby have less activity. B. Sunlight. Light helps herbicide penetration by stimulating stomatal opening and movement from the leaf to other parts of the plants by activating photosynthesis, leading to greater movement of organic solutes. Light is a prerequisite for the activity of all the herbicides since nearly 80% of the herbicides are affecting photosynthesis directly or indirectly. 305. 1239. C. Rainfall. Rain soon after application washes off the spray deposits of the foliage and therefore reduces herbicide effectiveness. However, the nature, amount, duration and distribution of rainfall may play a great role towards herbicides' activity and selectivity. Soil factors. I. Organic matter content. Organic matter having huge surface area and negative charges influences herbicide availability to plants. Higher the organic matter content, higher is the dose of herbicide required. Level of efficiency of a herbicide. Weak base herbicides, triazines, triazinones, triazoles, pyridinones, cashnic herbicides, bipyridiniums, and non-ionic polar and apolar herbicides undergo sorption with soil colloids and become unavailable to plants. This results in decreased activity of the herbicides. B. Herbicide and soil moisture or irrigation interaction. Adequate soil moisture in surface soil is necessary for germination of large population of weeds and their effective killing by herbicides. The adequate soil moisture increase the adsorption and translocation of soil applied as well as foliage applied herbicides which increase efficiency of herbicides in killing weeds. The excess moisture as well as soil moisture stress affect the absorption and efficiency of herbicides and heavy rains after application of herbicides may cause washing away of foliar applied herbicides. C. Texture and structure of soil. Heavily textured clayey soil requires more dose of herbicide compared to light textured sandy soil for the same degree of weed control. Leaching of herbicide is more in light soil, whereas adsorption is more in heavy soil. In light soil, leaching, therefore, may cause phytotoxicity and affect selectivity by reaching to the crop root zone even though the dose of herbicide is less. D. Soil pH. Generally, herbicides exist in ionic form between pH 4.3-7.5 with an increase in ionic concentration being proportional to an increase on pH. 306. 1239. Effects of herbicides. Effects of herbicides on environment. Herbicides vary greatly in chemical composition and in the degree of threat they pose to the environment. Many of the herbicide are highly persistent. It is widely recognized that main reason accounting for residue of certain herbicide like simazine and other triazines in ground and the surface water was the widespread use of decide that high doses on hard surface. Soil. Some herbicides are non-biodegradable and are harmful for a long period of time. Heavy dose of herbicides affect microbial population of the soil. With herbicides targeting amino acid synthesis in both plants and microbes, there is a possibility that N2 fixation may be inhibited by the application of certain herbicides. B. Water. The improper use of pesticides and herbicides may also cause the storm water infiltration into groundwater. When these pesticides and herbicides contaminants dissolve in storm water, they infiltrate the groundwater and then the surface waters, such as ponds, streams, rivers and lakes. These chemicals may also find their way into the soil and deeper groundwater units, polluting them. C. Living organisms. Most herbicides are specifically plant poisons and are not very toxic to animals. However, by changing the vegetation of treated sites, herbicide use also changes the habitat of birds, mammals, insects, and other animals through changes in the nature of their habitat. Herbivores may eat the plants treated with herbicides and then carnivores eat the herbivores. The toxic herbicide would be passed up the food chain, increasing in concentration each time, resulting in cancers and even deaths. Anxiety about chemical residues in the environment has increased greatly in the last decade. These fears and concern about possible litigation have led many land managers to reappraise their weed control strategies. Change has also been forced on them by the decrease in the number of approved herbicides as a result of the high cost of registration. In addition, approval has been withdrawn from more toxic and persistent herbicides. B. Effects of herbicides on humans. Among the many effects of pesticides and herbicides, perhaps the most alarming is the danger they pose to human health. People are directly affected by toxicity of some herbicides during the course of their occupation, i.e., when spraying pesticides, or indirectly affected when exposed through drift or residues on food and wildlife. Pesticides and herbicides can cause a number of health problems such as heart congestion, lung and kidney damage, low blood pressure, muscle damage, weight loss and adrenal glands damage. B. Arbitrary and indiscriminate usage of herbicides and pesticides can result in endometriosis, a common cause of infertility in women. 307. 1240. Herbicides and pesticides have been suspected by National Cancer Research Institute as a probable cause for certain cancer, that is cancer of that brain, prostate, stomach, lip, as well as leukemia, skin, melanoma, etc. Especially among farmers, the National Academy of Science reported that infant and children, because of their developing physiology, are susceptible to negative effect of herbicides and pesticides in comparison to adult. C. Effect of herbicides on crop. An important problem with broadcast application is that they are non-selective. They are toxic to a wide variety of plant species, and not just the weeds. If herbicides are not used properly, damage may be caused to crop plants, especially if too large dose is used, or if spraying occurs during a time when the crop species is sensitive to the herbicide. Unintended but economically important damage to crop plants is sometimes a consequence of the inappropriate use of herbicides. Is that they are the buildup of biotypes. Apart from their effect on the environment, another major problem with herbicides has been the buildup of herbicide-resistant biotypes where the same herbicide has been used repeatedly for a number of years. This problem was not clearly foreseen at the start of the herbicide revolution, but since the early 1980s, triazine resistance has developed in most countries where these herbicides have been used. The usefulness of a number of other herbicides, including paraquat dichlorphopmethyl and sulfonylurea types, has been affected by the development of resistant biotypes. Methods of dealing with this problem include prevention of weed seed shading, crop rotation, herbicide rotation, control of weed escapes, and tillage practices. Crop rotation is not relevant in an amenity situation where the crops are usually perennial, but other control measures may be appropriate in certain situations. If weeds are prevented from setting seed, resistant biotypes cannot develop. This could be achieved if land managers were made more aware of the threat of resistant biotypes and made greater efforts in intensively managed areas to prevent weeds from shading seeds by the use of a rotation of herbicides supplemented by physical means such as mulching, hand hoeing, and hand weeding. Modern, intensively managed agricultural and forestry systems have an intrinsic reliance on the use of herbicides and other pesticides. Unfortunately, the use of herbicides and other pesticides carries risks to humans through exposure to these potentially toxic chemicals, and to ecosystems through direct toxicity caused to non-target species, and through changes in habitat. Nevertheless, until newer and more pest-specific solutions to weed management problems are developed, there will be a continued reliance on herbicides in agriculture, forestry, and for other purposes, such as lawn care. 308. 1240. Management of herbicide residues in soil. An ideal sale-applied herbicide should persist long enough to give an acceptable period of weed control, but not so long that soil residues after crop harvest limit the nature of subsequent crops which can be grown. Various management techniques have been developed which can help to minimize the residue hazards in soil. A use of optimum dose of herbicide. Hazards from residues of herbicides can be minimized by the application of chemicals at the lowest dosage by which the desired weed control is achieved. Besides, applying herbicides in bands rather as broadcast will reduce the total amount of herbicide to be applied. This will be practicable in line sown crops or crops raised along ridges, such as cotton, sugarcane, sorghum, maize, etc. B. Application of farmyard manure. Farmyard manure application is an effective method to mitigate the residual toxicity of herbicides. The herbicide molecules get adsorbed in their colloidal fraction and make them unavailable for crops and weeds. Besides, FYM enhances the microbial activity, which in turn degrades the herbicide at a faster rate. C. Plowing. Cultivating the land. Plowing with disc plow or intercultivators reduces the herbicide toxicity, as the applied herbicide is mixed to a large volume of soil and gets diluted. In case of deep plowing, the herbicide layer is inverted and buried in deeper layers, and thereby the residual toxicity got reduced. D. Crop rotation. Ragi cotton sorghum is the common crop rotation under irrigated field conditions of Coimbatore district. Flucloraline 0.9 kg or butachlor 0.75 kg plus hand weeding at 35 DAT for ragi sunflower, border crop. Pendimethalin 1.0 kg plus hand weeding on 35 DAS for cotton intercropped with onion, and 2 manual weeding at 15 and 35 DAS for sorghum intercropped with cowpea is the recommended weed control practice. The above weed management schedule did not show any residual effect in the cropping system because the herbicides are changed for every crop. Use of non-phytotoxic oil. Atrazine residual hazard could be reduced by mixing non-phytotoxic oil, which would also enhance the weed killing potency. F. Use of activated carbon. Activated carbon has a high adsorptive capacity because of its tremendous surface area, which vary from 600 to 1200 m. Superscript 2G. Incorporation of 50 kg of activated charcoal. Inactivated completely chlorsulfuron applied at 1.25 and 2.50 kg and did not affect the yield of match compared to 309. 1241. Untreated control application of charcoal at 5.0 kg along the seed line reduced the residual toxicity of atrazine in soybean crop. G. Use of safeners and antidotes. A new development in herbicide usage is the use of safeners and antidotes in order to protect the crop plant from possible damage by a herbicide. This means that it may be possible to use certain herbicides on crops that would normally be affected by herbicide. Na-1,8-naphthalic anhydride has been used as a seed dressing on rice to protect the crop against molinate and alachlor. Another herbicide, safener siamtrinil, is used along with metolchlor in grain sorghum and other crop species. H. Leaching the soil. Leaching the herbicide by frequent irrigation is possible, especially in case of water-soluble herbicides. In this case, the herbicides are leached down to lower layers beyond the reach of the crop roots. Herbicide resistance. Naturally occurring inheritable ability of some weed biotypes within a population to survive a herbicide treatment. Herbicide resistance, HR, is the inherited ability of a plant to survive and reproduce following exposure to a dose of herbicide normally lethal to the wild type. Senecio vulgaris resistance to triazine group of herbicide was noticed during 1970. In India, the most common example is Phalaris minor developed resistance against isoproteron. The highest resistance in 61 weed species was recorded for atrazine types of resistance 1. Multiple resistance is the phenomenon in which a weed is resistant to 2 or more herbicides having different mechanisms of action. An example would be a weed resistant to sulfonylurea herbicides, ALS inhibitors, and glycinase, EPSP synthase inhibitors. 2. Cross resistance occurs when the genetic trait that made the weed population resistant to one herbicide also makes it resistant to other herbicides with the same mechanism of action. An example would be a weed resistant to imidazolinone herbicides, ALS inhibitors, and sulfonylurea herbicides, also ALS inhibitors. 310. 1241. Mechanisms of herbicide resistance. 1. Altered target site. An herbicide has a specific site, target site of action, where it acts to disrupt a particular plant process or function. Mode of action. If this target site is somewhat altered, the herbicide no longer binds to the site of action and is unable to exert its phytotoxic effect. This is the most common mechanism of herbicide resistance. 2. Enhanced metabolism. Metabolism within the plant is one mechanism a plant uses to detoxify a foreign compound such as an herbicide. A weed with the ability to quickly degrade an herbicide can potentially inactivate it before it can reach its site of action within the plant. 3. Compartmentalization or sequestration. Some plants are capable of restricting the movement of foreign compounds, herbicides, within their cells or tissues to prevent the compounds from causing harmful effects. In this case, an herbicide may be inactivated either through binding, such as to a plant sugar molecule, or removed from metabolically active regions of the cell to inactive regions, the cell wall, for example, where it exerts no effect. 4. Overexpression of the target protein. If the target protein, on which the herbicide acts, can be produced in large quantities by the plant, then the effect of the herbicide becomes insignificant. Conditions favorable for development of herbicide resistance. 1. Herbicide application inadequate rate, poor spray coverage and or incorporation improper timing of application of post-emergence herbicides, after weeds are too large to control, failure to use an adjuvant, if needed. 2. Soil and or climatic conditions, excessively wet or dry soil, seedbed condition, clods, etc. Herbicide absorption to soil particles or organic matter, lack of timely rainfall for activation, wash off of post-emergence herbicides. 3. Repeated use of same herbicide or use of herbicide with same mode of action due to the practices of monoculture. 311. 1241. 4 areas where minimum zero tillage is followed. 5 fields where farmers rely on only herbicides for high degree level of weed control, including nurseries, orchards. 6. Non-crop situations like roadsides, railway tracks, etc., where herbicides are repeated used may be at higher doses than crop situation. Strategies for preventing or managing herbicide resistance. Practice crop rotation. Rotate herbicide families and use herbicides with different modes of action. Use herbicide mixtures with different modes of action. Control weedy escapes and practice good sanitation to prevent the spread of resistant weeds. Integrate cultural, mechanical, and chemical weed control methods. Recognizing weed characteristics that promote resistance, and annual weeds can have several reproductive generations per growing season. Cross-pollinate tend to be more prone to develop resistance. Species that have seed dispersal over greater distances also tend to be more prone to develop resistance. 312. 1241. 7 recent developments in weed management. 1. Allelochemicals. Allelopathy is a biological phenomenon by which an organism produces one or more biochemicals that influence the growth, survival, and reproduction of other organisms. These biochemicals are known as allelochemicals and can have beneficial, positive allelopathy, or detrimental, negative allelopathy, effects on the target organisms. Allelochemicals are a subset of secondary metabolites, which are not required for metabolism, ge growth, development and reproduction, of the allelopathic organism. The term allelopathy or teletoxy was introduced by Molist, 1937. Allelochemicals are found to be released to environment in appreciable quantities via root exudates, leaf leachates, roots and other degrading plant residues, which include a wide range of phenolic acids such as benzoic, 1, and cinnamic acids, 2, alkaloids, 3, terpenoids, 4, and others. These compounds are known to modify growth, development of plants, including germination and early seedling growth. There are increasing evidences that allelochemicals have significant effects on cell division, cell differentiation, ion and water uptake, water status, phytohormone metabolism, respiration, photosynthesis, enzyme function, signal transduction as well as gene expression. Selected allelochemicals. 1. Benzoquinones. Sorgoleone and its hydroquinone are allelochemicals exuded from the roots of form sorghum. Sorgoleone is highly phytotoxic and inhibits chlorophyll formation, photosynthetic oxygen evolution. 2. Coumarins and flavonoids. Coumarins and flavonoids are ubiquitous in plants, and several have been implicated in allelopathic interactions. 3. Terpenoids. Mesogenic acids are a family of 9 lupane triterpenes isolated from sweet clover. 4. Strigolactones. Parasitic weeds of the striga, witchweeds, arrowbunch, and alectra families affect a number of important cereal and legume crops, causing dramatic reduction in yield and in severe cases complete destruction of the crop. 313. 1242. Helianols. The helianols are a promising group of phenolic allelochemicals isolated from H. annuus. Unicals. Allelopathic control of certain weeds using botanicals. For instance, dry dodder powder has been found to inhibit the growth of water hyacinth and eventually kill the weed. Likewise, carrot gross powder found to detrimental to other aquatic weeds. The presence of marigold, Tagetes erecta, plants exerted adverse allelopathic effect on Parthenium spp growth. The eucalyptus tree leaf leachates have been shown to suppress the growth of nutsedge and Bermuda grass. 2. Bioherbicides. In irrigated agriculture, weed control through chemical herbicides creates spray drift hazards and adversely affects the environment. Besides, pesticide residues, herbicides, in food commodities, directly or indirectly affect human health. These led to the search for an alternate method of weed management, which is eco-friendly. In this regard, the biological approach, a deliberate use of natural enemies to suppress the growth or reduce the population of the weed species, is gaining momentum. This approach involves two strategies, the classical or inoculative strategy, and the inundative or bioherbicide strategy. In the inoculative approach, an exotic biocontrol agent is introduced in an infested area. This method is slow and is dependent on favorable ecological conditions, which limits its success in intensive agriculture. Whereas in the inundative approach, bioherbicides are employed to control indigenous weed species with native pathogen, applying them in massive doses in the area infested with target weed flora. A bioherbicide is a biologically based control agent for weeds. Bioherbicides are made up of microorganisms, e.g. bacteria, viruses, fungi, and certain insects, e.g. parasitic wasps, painted lady butterfly, that can target very specific weeds. The microbes possess invasive genes that can attack the defense genes of the weeds, thereby killing it. Bioherbicides may be compounds derived from microbes such as fungi, bacteria or protozoa, or phytotoxic plant residues, extracts or single compounds derived from other plant species. A bioherbicide based on a fungus is called a mycoherbicide. In the industry, bioherbicides and other biopesticides are often referred to as naturals. A bioherbicide is a preparation of living inoculum of a plant pathogen, formulated and applied. 314. 1242. In a manner analogous to that of a chemical herbicide in an effort to control or suppress the growth of weed species. Commercial bioherbicides first appeared in the market in USA in early 1980s with the release of the products Devine, Coligo and Biomil. Devine, developed by Abbott Laboratories USA, the first mycoherbicide derived from Unk, Phytophthora palmivora, is a facultative parasite that produces lethal root and collar rot of its host plant Morenia odorata, Stangler vine, and persists in soil saprophytically for extended periods of residual control. It was the first product to be fully registered as a mycoherbicide. Commercially, Coligo, a formulation of endemic anthrocnous fungus Colectotrichum glyspirioides F. sp. Esquimone, was developed to control northern joint wedge, Esquimone virginica, in rice and soybean field. The successful development of Coligo led to the discovery of another Colectotrichum-based mycoherbicide, Biomil, by Philome Bios Inc., Canada. It contains spores of C. gliosporioides says TSP. Mulway. It is used to control Mahapusilla, round-leaved mallow, in Canada and USA. Bioherbicides offer many advantages. They include a high degree of specificity of target weed, no effect on non-target and beneficial plants or man, absence of weed resistance development, and absence of residue buildup in the environment. Recently, the potential of many microorganisms, especially fungus, to control weeds in several crops has been reported. Some of them are listed here. Cercospora rhodmani, ABG 5003, Cercospora cocodes, Whalego, Fusarium aeonifloral deleterious rhizobacteria, DRB. Pseudomonas spp agrobacterium xanthomonas spp irvinia herbicola. Limitations in use of bioherbicides. Besides many advantages of bioherbicides, certain factors have been reported to limit the development of bioherbicides into commercial products. These include biological constraints, host variability, host range resistance mechanisms, and interaction with other microorganisms that affect efficacy. Environment constraints, epidemiology of bioherbicides dependent on optimum environmental conditions. 315. kanagrajbioscience@gmail.com. 1243. Technical constraints, mass production and formulations, development of reliable and efficacious biocide, and commercial limitations, market size, patent protection, secrecy and regulations. The bioherbicides approach is gaining momentum. New bioherbicides will find place in irrigated lands, wastelands, as well as in mimic parasite weeds or resistant weed control. Research on synergy test of pathogens and pesticides for inclusion in IPM, developmental technology, fungal toxins, and application of biotechnology, especially genetic engineering, is required. However, bioherbicides should not be viewed as a total replacement to chemicals, but rather as complementary in integrated weed management systems. 3. Weed management in conservation agriculture systems. Conservation agriculture, CA, is defined as a sustainable agriculture production system comprising a set of farming practices adapted to the requirements of crops and local conditions of each region, whose farming and soil management techniques protect the soil from erosion and degradation, improve its quality and biodiversity, and contribute to the preservation of the natural resources, water and air, while optimizing yields. Agronomic practices included in CA are based on 3 core principles, which must be fulfilled concomitantly. 1. Minimum soil disturbance. 2. Maintenance of permanent soil covers. 3. Cropping system diversity, crop rotations. CA in India. Times reduced costs, increased profitability and better use of resources, e.g., labor and water, are the main factors responsible for the adoption of CA in India. Impending changes in climate and sustainability of cropping systems are also important reasons for adoption of CA systems in India. In the Indo-Gangetic Plains, the resource conservation technologies are being practiced in 3 million ha under the rice wheat-based systems. Challenges. Despite several benefits CA offers, weeds continue to be one of the biggest constraints to its adoption. After adoption of zero-till wheat in northwest India, weed flora shifted towards broadleaf weeds including toothed dog, Rumex dentatus L. Similarly, in the vertisols of Jabalpur, infestation of common wedge, garden wedge, Vishya sativa L. increased in zero-till system compared to conventional tillage system. 316. 1243. As adoption of CA practices has been increasing, there is a need to gain better understanding on weed management in CA crop production systems. Various approaches. Approaches including the use of preventive measures, intercropping, cover cropping, crop residue as mulches, competitive crop cultivars, optimum planting geometry, optimum sowing time, herbicide-tolerant, HT, cultivars and herbicides, components of integrated method, need to be followed to successfully manage weeds. The use of clean crop seeds and clean machine, which are much cheaper and easier options in reducing weedy rice infestation in CA systems. In CA system, crop residues are left behind on the soil surface. In addition to moisture and soil conservation, the residues act as mulch and suppress weed seedling emergence. Inclusion of a cover crop between two main crops also helps reduce weed density in CA cropping system. In this, the cover crop can be killed by using a non-selective herbicide and its dead mulch be used to suppress weed germination by releasing allelochemicals and or reducing light transmittance to soil surface. Growing Sesbania rostrata as a cover crop was found to control most of the weeds, leaving the field almost weed-free in rice wheat cropping systems. Similarly, mung bean can be grown as a cover crop in rice wheat cropping system. Crop rotations and diversification not only improve soil health but also reduce buildup of pests, including weeds. Different crops require different management programs to prevent selection of resistant weed species in CA systems. In India, fewer cases of resistance in Phalaris minor were found when growers included sunflower and sugarcane in rotation than following a continuous rice wheat cropping system. Herbicides play an important part in managing weeds in CA. However, due to presence of crop residues on the soil surface, pre-emergence herbicides may not be very effective. Residues are known to intercept up to 80% of the applied pre-emergence herbicides. Therefore, there is a need to better understand the efficacy of different pre-emergence herbicides when applied in different crops in India. Because of the efficacy issue of pre-emergence herbicides, timing of post-emergence herbicides is critical in CA. Herbicide rotations and mixtures may improve the weed control spectrum. Times there is a need to integrate different weed management strategies for widening the weed control spectrum and maintaining the sustainability of CA. 317. 1243. 4 transgenic herbicide-resistant crops. Herbicide resistance is the major trait that has been engineered into crops, and herbicide-resistant crops, HRCs, occupy the largest area under transgenic crops in the world. The problem to weed resistance to herbicides has provided an invaluable opportunity for visionary scientists to develop crops resistant to previous selective herbicides. Researchers explored ways to develop crops resistance to herbicides with the goals of improving herbicide selectivity, expanding weed control spectrum, as well as minimizing crop injury. HRCs can be classified as non-transgenic, traditional genetic methods of selection of resistance traits, and transgenic, genetically engineered. Non-transgenic HRCs were developed using conventional breeding techniques. The first such example is triazine-resistant canola that was developed through a breeding program in 1984. Transgenic herbicide-resistant crops. The initial efforts to develop transgenic HRCs using genetic engineering techniques resulted in the release of bromoxynil-resistant cotton in 1995 and canola in 2000. Bromoxynil is not a broad-spectrum herbicide, therefore, bromoxynil-resistant cotton and canola were discontinued. The real turning point occurred in 1996-97 with the commercial release of glyphosate-resistant, GR, canola, soybean and cotton. These crops allowed the application of glyphosate multiple times in the growing season without risk of crop injury. Current transgenic resistant crops and associated trait genes. Alfalfa, cotton, soybean, sugar beet, glyphosate CP4 EPS, agrobacterium strain called CP4 resistant to glyphosate. Tolerance to the herbicide glufosinate, BASTA, is conferred by the bacterial gene BAR, which metabolizes the herbicide into a non-toxic compound. Glyphosate, another most popular herbicide, resistance is achieved by the introduction of either agrobacterium gene CP4 that codes for a glyphosate-insensitive version of the plant enzyme, EPSP synthase, or GOX gene from Achromobacter, which codes for glyphosate oxidoductase in the breakdown of glyphosate. Potential impacts, risks and benefits of HRCs and the herbicides that are used with them. The only herbicides currently being used with HRCs, glyphosate and glufosinate, are more environmentally and toxicological benign than many of the herbicides that they replace. The effects on soil, air and water contamination and on non-target organisms are relatively small. 318. 1244. Soil erosion causes long-term environmental damage being broad spectrum. Foliarly applied herbicides, with little or no activity in soil, glyphosate or glufosinate are highly compatible with reduced or no-tillage agriculture and have contributed to the adoption of these practices in the Western Hemisphere. This contribution to environmental quality by HRCs is perhaps the most significant one. Transgenic foods are tested and evaluated for safety to a much greater extent than traditional foods that are usually derived by conventional breeding methods involving the transfer of many genes. New crops and new crop varieties of conventional crops are usually introduced without any testing for safety or nutritional problems related to genetics. In contrast, a rigorous safety or nutritional problems related to genetics. In contrast, a rigorous safety testing paradigm has been developed for transgenic crops which utilize a systematic, stepwise and holistic approach. Foods and feeds derived from genetically modified crops are as safe and nutritious as those derived from traditional crops. The lack of any adverse effects resulting from the consumption of transgenic crops grown on hundreds of millions of cumulative hectares over the last 15 years supports this conclusion. High levels of adoption of HRCS have dramatically increased the use of herbicides, with a following increase in resistant weeds and weed species shifts. Management of resistance weeds requires alternative strategies that should not rely solely on different herbicide mechanism of action. HRCS and other transgenic crops are here to stay. While the benefit of these crops are plentiful, there are certain inherent consequences, some obvious and a few quite apparent. The most important issue requiring attention following commercialization of HRCS has been the evolution of resistance to herbicides in weed populations. The agrochemical industry, seed companies and related entities have invested most of their resources in development of the next generation of HRCS and other transgenic crops, with the aim of diversifying the grower's crop portfolio as well as combating weed resistance by providing cropping technologies that allow application of more than one mode of action herbicides. 5. Management of problem, perennial and parasitic weeds. I. Cynodon dactylon and Cyperus rotundus. Management of perennial weeds like Cynodon dactylon and Cyperus rotundus by the application of glyphosate 15 ml plus ammonium sulfate 20G plus activator 1 ml of water. 319. 1244. 2. Partanium hysterophorus manual removal and destruction of Partanium plants before flowering using hand gloves machineries, or pre-emergence application of atrazine 4G liter in 500 liters of water hectare, or uniform spraying of sodium chloride 200G plus 2 milliliters soap oil liter of water, or post-emergence application of metribuzin 3G liter of water under non-crop situation raising competitive plants like Cassia sericea and Abutilon indicum on fallow lands to replace Partanium, or biological control by Mexican beetle which is very active during only in monsoon seasons. 3. Ipamea carnea foliar application of 2,4-D sodium salt 8G plus urea 20G soap oil 2 ml liter of water and then removal and burning of dried weeds, or manual mechanical removal of grown-up plants in channels during summer. 4. Achonia crassipes manual mechanical removal and drying application of 2,4-D sodium salt 8G plus urea 20G or paraquat 6 ml liter of water application of glyphosate 15 ml plus ammonium sulfate 20G liter of water V. Striga asiatica times pre-emergence application of atrazine 1.0 kg ha on 3rd dab plus hand weeding on 45 dab with an earthing up on 60 dab combined with post-emergence spraying of 2,4-D 6G, 0.6%, urea 20G, 2%, liter of water on 90 dab plus trash mulching 5T ha on 120 dab VL or bunch times. Pre-emergence application of pendimethalin 1.0 kg ha or oxyflurfen 0.30 kg ha on 3 dab in tobacco, tomato and brinjal and 3 dab in potato. Times plant hole application of neem cake 25G plant or drenching of copper sulfate 5% provides partial control of or bunch in tobacco. Times directed application of paraquat 6 ml liter of water or glyphosate 8 ml liter of water or imidacloprid 3 ml liter of water on the or bunch shoots. 320.